Okay, we're going to review brain structures on this model, and then we'll take a look at the structure of the meninges, and we'll also look at the ventricular system and talk about the flow of cerebrospinal fluid on this model. Okay, so just to remind us, this is the cerebrum in here. Here's the corpus callosum. Okay, here's the diencephalon, thalamus, hypothalamus, epithalamus. Here's the brainstem, midbrain, pons, medulla, and here's the cerebellum. Okay. All right, so first let's look at the meninges. Okay, we have the same three types of meningeal layers surrounding the brain as we have surrounding the spinal cord. The dura mater, the arachnoid mater, and the pia mater. Okay, on this model, the dura is shown in gray. The arachnoid, the membranous part of the arachnoid, is shown as this orange line. Okay, and you can see it here as well and over here. Okay, and the pia, or pia mater, is shown as the red line. Okay, so that red line is the pia mater. Okay, so the dura is the one closest to the skull, the internal surface of the skull. The pia is the layer closest to the brain tissue, and the arachnoid is what's in between. Okay, and remember that within the arachnoid we have a space. Okay, so we have this membranous part of the arachnoid that's shown with the orange, but then there should be webbing going across here, and we don't really see the webbing because it's really hard to represent on these models. But where that webbing is also where we have the subarachnoid space. And that's the space that's filled with cerebrospinal fluid. And in just a minute we'll talk about how that fluid gets to this space. Okay? Now, one of the differences between the meninges in the cranial cavity compared to those around the spinal cord is that the dura actually has two layers here in the cranial cavity. The layer of the dura that is associated with the spinal cord becomes the meningeal dura in the cranial cavity. Okay, but because we want to have a close association between the meninges and the skull, the internal surface of the skull, the periosteum of the bones that form the cranial cavity is going to be our other layer of the dura. Okay, so we have the meningeal dura that's continuous with the dura of the spinal cord, and then we have the periosteal dura, which is what is really the periosteum on the internal surface of our cranial bones. Now, in most places, like right here, those two layers are fused with each other. So you can see there's no separation between them. In other areas, though, the layers are separated, like here also. Okay. Now, in those places where we have a separation between the two layers of the dura, we form channels where blood can collect. And this is venous blood that's draining away from the brain. Okay, and all of these channels, which are referred to as dural sinuses, eventually lead to a central location that feeds into the internal jugular vein. Okay? Now, we talked about the venous component. So let's talk about the arterial component a little bit. There are a couple of sets of arteries, and you'll learn more about those in A&P2, that are bringing oxygen-rich blood to the neurons in the brain. And remember, neurons are very highly active metabolically, need that ready blood supply. Okay, now eventually we'll get through the different sizes of blood vessels to the brain tissue, and we'll get the blood into capillaries. Most of the capillaries are going to be associated with providing oxygen and nutrients and picking up waste from the neurons. But there's a special set of capillaries that produce the cerebrospinal fluid. And these capillaries are called the choroid plexus. Okay, every one of the spaces inside the brain, large spaces that is, called ventricles, is going to have a set of these choroid plexus capillaries within it. Okay, and as the blood comes in, there's a filtration process that occurs, and basically the plasma of the blood, at least a subsection of it, subdivision of it, is allowed to move through these capillaries into these spaces. And that fluid that is filtered by the choroid plexus becomes the cerebrospinal fluid. Okay, so I'm turning this model around, and as we look at it this way, now we're seeing a more lateral section through the cerebrum, okay, and through the cerebellum as well. Okay, and as we're looking at the cerebrum, we can see that there's this big kind of C-shaped structure inside the cerebrum, and that is one of the lateral ventricles. Each cerebral hemisphere has a lateral ventricle within it. Okay, and this would be the choroid plexus that would be producing cerebrospinal fluid and allowing it to move into this ventricle. Okay, now from here, this cerebrospinal fluid will move through this opening, which is the interventricular foramen, to come into the third ventricle. Here it will mix with the cerebrospinal fluid that's coming from the other cerebral hemisphere, so from the other lateral ventricle. And it will also mix with cerebrospinal fluid that's being produced in this space, because this is choroid plexus back here as well. So this space that all of these batches of cerebrospinal fluid are mixing in, this is called the third ventricle. Okay, so there's no first and second ventricle. That confuses people sometimes. The two ventricles in the cerebral hemispheres, which are the lateral ventricles, you could think of as the first and second. But this one is considered the third. Okay, so all of that cerebrospinal fluid from the two lateral ventricles and from the third ventricle will pass through this narrow channel, which is the cerebral aqueduct. That goes through the midbrain, and eventually get to this ventricle, which is the fourth ventricle. And there it will mix with cerebrospinal fluid that's being produced in this space as well, because here's some more choroid plexus. Okay, now the majority of the cerebrospinal fluid is then going to go through openings to get to the subarachnoid space. So remember I told you that I would tell you how we got the fluid into the subarachnoid space? This is how. There are openings called apertures in the fourth ventricle. There are two lateral apertures, which you can't really see here. There's also a median aperture, which you also really can't see here on this model. Okay? But those allow the cerebrospinal fluid to move into the subarachnoid space. Okay, and that's where most of it goes. And this is what gives us that layer of fluid that buffers the brain and protects it by preventing it from hitting against the skull internally as we're moving around. Okay, a small percentage of the fluid will also go into the central canal of the spinal cord. Okay, and some will also circulate around in the subarachnoid space around the spinal cord. Okay, now remember that this fluid came from the blood one of the truths in anatomy and physiology is any fluid that comes from the blood eventually has to return to the blood. Okay, because your blood volume has to stay the same all the time. So different systems borrow fluid from the blood, but they always have to give it back. You know, that's the thing with borrowing. You get it for a while, but you have to give it back. Okay? So how do we give this fluid back to the blood? Well, we have places where the arachnoid extends through the dura, and these little locations act as one-way valves. These are referred to as arachnoid villi. And this little orange bump that you see here is one. Okay, so the fluid is circulating in the subarachnoid space. It gets to the arachnoid villus. It goes through that valve, and then here it is in the venous blood. Okay, so it came from capillaries, circulated in the ventricles, and then through the subarachnoid space. It's now coming back to the blood in these dural sinuses. Okay, and so the blood leaving the cranial cavity is going to be basically the same volume as the blood entering the cranial cavity. Okay, now here's another representation of the ventricles. Okay, we'll do a lateral view of this first. This is where you can see the lateral ventricle the best. You can see it's roughly C-shaped. Okay, and there's one on each side. Okay, and here's an anterior view. Okay, so this one would be, this lateral ventricle, would it be inside the right cerebral hemisphere? This lateral ventricle would be inside the left cerebral hemisphere. And you can see there's a little stalk on each of these. Okay, and I should tell you, this is just a representation of the ventricular system. So as if you filled up the ventricles with something solid and then tore all the brain tissue away. This is what the ventricles would look like by themselves. Okay, so the two lateral ventricles, each of these little stalks that connects the lateral ventricle to the mid piece here, that would be an interventricular foramen. So there's one here and one over here. Those both lead to, turn it back this way, the third ventricle. And the third ventricle is the one that always has the hole in it. And that hole is there because there's actually a structure that runs through there that connects the two halves of the thalamus. Okay, and this is the little piece that goes down into the hypothalamus. You know how the hypothalamus kind of looks like a W at the edge, or it has that kind of toothed look. And then you look at this part of the third ventricle and see how it has that same kind of shape. Okay, so this is the part in the hypothalamus, the third ventricle section of the hypothalamus. Okay, and then we have this little narrow section here. This is the cerebral aqueduct. And then the last section, I'll turn it around again, is the fourth ventricle. Okay, and right here is where the median aperture would be, and here are where the lateral apertures would be. Where you see pink on this model, that's where the choroid plexus is. Okay, so you can see there's pink, you get that? Pink in each of the lateral ventricles. Okay, and then there's also pink in the third ventricle and in the fourth ventricle. So every ventricle has choroid plexus in it, and every one makes cerebrospinal fluid.